OK, thanks everyone for being there. Merci tout le monde d'être là à la conférence de l'IBIS. Aujourd'hui, on a la chance d'accueillir Dr. Kin Chan, qui est professeur à l'Université d'Ottawa, à la faculté de médecine. Son labo, il a commencé sa carrière de chercheur indépendant en 2016 à l'Université d'Ottawa. Donc, c'est relativement un nouveau labo. Et il travaille en fait, quelque chose de très fondamental en biologie, mais avec une application en biologie médicale aussi, qui est l'étude des patrons et des taux de mutation. Donc, c'est de ça dont il va nous parler aujourd'hui. Et sa formation est multidisciplinaire. Il a commencé comme un étudiant en biologie à Caltech, où il a fait son bac, puis où il a fait un peu de travail sur les récepteurs olfactifs chez l'humain. Puis ensuite, il a fait son doctorat à l'Université de Washington, à Seattle, où il a travaillé sur la physiologie de la levure avec Mark Roth. Puis, son postdoc, c'est là où il a commencé à faire plus de génomique et de bioinformatique. Et son postdoc s'est déroulé au NIH, où il a commencé à travailler sur des sujets qui s'apparentent à ce qu'il fait présentement. Donc, ses travaux présentement sont financés par le CRSMG, entre autres. Il détient aussi une chaire de recherche du Canada depuis son arrivée à Ottawa. Puis, il a aussi été financé pendant ses travaux aux États-Unis par le NIH, entre autres par des fonds qui aident les chercheurs à devenir indépendants et établir leur propre laboratoire. J'ai noté dans son CV aussi quelque chose d'intéressant qui s'implique dans le scientific outreach. Donc, il a fait des activités de vulgarisation, par exemple, dans plusieurs écoles à Ottawa, ce qui est intéressant, surtout considérant le sujet sur lequel il travaille. Donc, c'est beau de voir ces activités-là dans un CV pour un chercheur aussi avancé que lui. Donc, thanks so much, Dr. Chan, for coming, and we're looking forward for your talk.
Okay, well, thank you very much, Christian, for the invitation, and thank you all for taking the time to attend the webinar. So let's get going here. Let me share screen and pull up my slides. Okay, great. So I'm going to shrink the Zoom display. Okay, right. So yeah, so my talk today is going to look at base substitution patterns in different species, and hopefully I can convince you that there are deep links to what's going on in mutagenesis in cancers, which is in turn, we think, linked directly to energy metabolism. So turn on my pointer. Okay, right. So the talk is going to be subdivided into 3 parts. So first part is an introduction to mutational signatures in cancers. Second part is looking at the intrinsic base substitution patterns among different species. And then finally, there'll be some results with a yeast model system that we work with. Okay, so an introduction to mutational signatures. Okay, so to start with in a very broad way, as you're probably well aware, there are key enabling characteristics for cancers. And so this is taken from Hanahan and Weinberg. And so one of these key characteristics is genome instability and mutation. So at the beginning of this work, I was of the firm belief that basically genome instability underlies everything else that's happening in cancers. But as you'll see as we go through this talk, my view on this has started to shift a bit because of what we're finding. So in terms of genome instability in cancers, here's a sense of the scale of the question. So shown here are about 25 cancer types On the vertical axis is the frequency of somatic mutations per megabase of genome. So you can see immediately that this spans about 6 orders of magnitude or so. So on the low end, there are very sparsely mutated childhood cancers. And then on the high end, you have very heavily mutated cancers from mutagenic exposures. These are melanomas, so skin cancers, and also lung cancers. Within each type of cancer, you can also see that there's a broad range in terms of numbers of mutations per cancer genome. But this kind of frames the scale of mutagenesis that can occur in cancers. Now, on the bottom bar here, we see a representation of the mutational spectrum that is observed in these cancer types. So I should say that so each of these blocks is actually a multi-bar graph. So each of the bars represents one cancer genome. So you can see that there's, depending on the cancer type, the spectrum of mutations can vary quite a bit. Okay. So how do we make sense of all this? How do we find patterns within all this? Well, it turns out that a method called non-negative matrix factorization can be adopted to try to figure out what are the underlying recurrent patterns of mutation. So suppose we start out with a catalog of multiple cancer genomes, and they have different mutation types. What this NMF algorithm can do is essentially deconvolve this data set into putative recurrent patterns called mutational signatures and infer the contribution of each signature in the mutagenesis within each genome. So you end up with a set of putative signatures, estimations of the fractional contribution of each signature, and an error term, because the computational reconstruction of the original data set, it's never 100% perfect, so you always have an error term. Now, to get into this a little bit more, and I promise you this is the only equation in the entire presentation. So we can think of this as, pretty straightforward matrix multiplication and the inverse operation. So here we're representing our genomes, so genome 1 through genome G, and we're representing the different mutation types from type 1 in the subscript to type K. So that's our matrix of input data. The algorithm tries to deconvolve this into these patterns. So these are the mutational signatures. So they're expressed as probabilities. So that's what these P stand for. So the probability of observing a particular mutation type. And there are multiple putative signatures, so n putative signatures in this particular matrix. And over here in this E matrix, this E stands for exposure. So the implicit supposition is that each mutational signature is due to a specific mutagenic exposure, hence E for exposure. And so you would have multiple exposure values. So we have n putative signatures, so we have n possible exposure values to each signature, spanning all genomes in the data set, so from genome 1 through genome G. So yeah, so this is summarized here. So counts, start out with counts, set of putative signatures, and essentially the contribution of each signature. So when you run this in cancer data, what do you get? Well, you get signatures that look like this. So on the vertical axis is the percentage of single base substitutions. So So basically, this is the frequency of mutations. And on the horizontal axis, if we look at a larger view, so on the horizontal axis, on this fine scale, are trinucleotide motifs. So one base flanking the mutated base on the 5' side and one base flanking on the 3' side. So you end up with these 96 bar graphs. So for this signature, this is signature 1. It results from spontaneous deamination of 5-methylcytosine into thymine. And as you would expect, this happens in C to T results in C to T substitutions that occur in CPG motifs, so ACG, CCG, etc. And this is observed in all cancer types. Okay, so some further examples of prominent signatures that occur in cancers are signatures 2 and 13, which are basically two sides of the same coin. These result from the action of apobex-cytidine deaminases. So the apobex day job, essentially, is to restrict viruses by hypermutating them at cytosines. It turns out in many cancer types, the apobex deaminases, they essentially become deregulated and they start attacking genomic DNA. And so they leave behind these signatures. So signature 2 is C to T at TC motifs. And signature 13 is mostly C to G, also at TC motifs. Okay, so another prominent signature is number 3. This is from homologous recombination defects. And you can see this is a signature that doesn't have particularly high peaks. Instead, you get a mix of many, many different substitution types, each of them at relatively low frequency. So the scale here maxes out at just 3.3%. So you get a very, very mixed variety of substitution types. Okay, 2 signatures related to tobacco use, and these are number 4 and number 29. And you can see immediately they're actually quite similar. So what distinguishes them are more subtle details. So for both of them, most of the substitutions are C to A, and this is thought to be from oxidation of guanine. But you can see that the patterns aren't exactly the same. They aren't exactly superimposable. And in addition, for signature 4, there are other contributions. For example, this C to A contribution, which isn't observed in signature 29. And also they're related to different cancer types. So signature 4 is found in multiple cancer types, where signature 29 is just restricted to oral cancers. Okay, and final example here is signature 7 from ultraviolet light. So most of the mutations from signature 7 are, again, C to T substitutions, but specifically at dipyrimidine motifs. So these would be TC motifs. Typically, it could be CC motifs. And these are found in lip and skin cancers because they're sun exposed. Okay. So that was just a brief survey of some of the more prominent signatures. So as of right now, as of 2020, there are over 50 mutational signatures that have been identified with at least reasonably high confidence in cancer genomic data. And so, so this is the set, depending on who you ask. Some people say, actually, there are more, but others say, well, some of these may not be fully validated, and so we shouldn't include them. But the rough ballpark figure is about 50 or so, let's say. All right, so here's a figure showing the prevalence of these signatures in different cancers. So there's a lot of information on this slide. So let's walk through this systematically. So on the vertical axis here are the different signature types. Okay, so we have about almost 50 on this figure. And on the horizontal axis, there are about 30 cancer types. So the presence of a signature is indicated by one of these circles. And the size of the circle indicates the fraction of the cancer type that actually has the signature. Okay, so you have small circles if you have low proportion of cancer types with the signature, and then large circles for high proportion cancer types with the signature. And further, the coloration, the colors span from this yellowish to a deep purple. And so the purple is the high end. So the most highly, so the highest prevalence is indicated by the darker colors here. So you can see that of the different cancers, of the different signatures we looked at. So we looked at signature 1, it's found in all cancers. Signature 2 and 13, they're found in 24 cancer types, and so on and so forth. Okay. Right, so this doesn't have to do directly with signatures, but this is just a brief aside because this question actually comes up a lot. So folks have a question basically, well, why are there signatures? Why aren't all bases equally mutable? And that has to do with a couple of factors. So one is that there are different chemical moieties in the DNA. So for example, we have different amine groups, primary, secondary amines. We have ring nitrogens. We have ring carbons. Some of them are especially labile. So that accounts for some of the difference. And also, it turns out nearest neighbor differences can really tell depending on the type of DNA damage. And that's why when we look at, for example, the C to T block, all the peaks aren't equal height. Okay, so yeah, so that's going to be our brief introduction to mutational signatures. So let's go. Oh, by the way, if there are any questions that come up, if I say something that's unclear, please turn on your mic and ask a question. All right. Right. Okay, great. All right, so on to part 2, intrinsic-based substitution patterns. Okay, so when we started out with this, we just had a very simple question. So I tried to convince a student to work with some genomics data and just see, okay, what mutational patterns are in different species. I thought it would be an interesting project. At the time, we didn't know where it would go. I was not able to convince the student to do it, and so I ended up just started working with it on my spare time. So the way we started out is we just I downloaded a bunch of data. And so here are the data for 7 model species. So we thought, okay, we're a model species lab. Let's look at model species. So here are the different species, number of genomes that were sequenced by these authors. And all in all, we ended up with almost 800 million single nucleotide variants to look at. So it's pretty big data set. And if we were to just take these data from the 7 model species and plot them, sorry, it's hard to see because of the Zoom interface, but okay, yeah, okay. So if we plot them according to the mutational signatures convention, so C to A, C to G, C to T, so on and so forth, and then the trinucleotide motifs, then we end up with patterns that look like this. And what you can see immediately is that the C to T and the T to C, as a general rule, tends to outnumber the other categories. And this is, this is fairly consistent, although for some of the invertebrate species, for some reason, there are these high T to A peaks. We don't really know why, but they are there. Okay. So we could ask the question, okay, when we compare these to known signatures in the cancer database, are there any matches? And so to do this, we compute a quantity called cosine similarity. So here's a heat map showing cosine similarity, yellow on the low end, dark blue on the high end. So what you need to know about cosine similarity is that when two patterns are perfectly matching, the cosine similarity equals 1. So if you start getting values that are, say, 0.9 and above, then that indicates, okay, probably you have some sort of match. Because if you go through simulations and you figure out, okay, how likely is it to get a cosine similarity of 0.9 and above just by random chance, it turns out the chances of getting that are actually quite low. So on the order of 10 to the minus 6, something like that, if not lower. So in any case, taking these 7 sets of data, we computed the cosine similarities, and we found that they tend to match this signature 5. So, the text is hard to see, so magnified view. So here is cosmic signature 5, so mutational signature 5 from cancers. And so these are the darkest blue boxes. So that suggested to us, okay, maybe this is related to cancer signature 5 for some reason. All right. So I showed this to colleagues. And of course, being good scientists, they were skeptical or like, okay, well, you need more data. How do you know this isn't some sort of, fluky thing? So if you compute a consensus for the 7 model species, you end up with something like this. So these are 95% confidence intervals. And if you take the consensus and compare it to cosmic 5, you end up indeed with cosine similarity above 0.9. It's at 0.928. So so to look into this further, we downloaded all the available NCBI SNP data we could find. And this ended up being something like 600 million SNPs from over 40 species. We had to throw some of the species out because there were some obvious issues with some data sets because they're missing entire blocks of substitution types. But in any case, here are some of the species, and just quickly scroll through this. But what I want you to take away from this is that for most of these species, it's the orange blocks and the green blocks that tend to have the highest levels. Yeah, let's see. Yeah, okay, so that's the last one. Okay, again, comparing to the cancer mutational signatures. So here are all our species from the NCBI SNP database. And you can see there's a range of different kingdoms even. So there are many vertebrates, there are some plants mixed in there, some invertebrates as well. We just took on all comers. So again, the cosine similarity, you see most of the dark blue is right here in this column, which is again signature 5 from the cancer database. And again, we computed the consensus for 41 species, not including human. And so this is a consensus with 95% confidence. So comparing the consensus SNP pattern to the model systems consensus, we get cosine similarity of over 0.98. So this is pretty close match as far as these things go. The consensus SNP pattern compared to the cancer signature, cosmic signature 5, we get cosine similarity of 0.931. And then finally, we had the thought, okay, what about human SNPs? would we see something similar? And so we looked at over 500 million human SNPs. And so human SNPs versus cancer signature 5, and the cosine similarity is 0.957. So we think the mutagenic mechanisms for SNPs and for somatic mutations are probably the same thing. Okay, so all this stuff about signature 5, what's known about signature 5? If we revisit this chart, here is signature 5. So it's present in all cancer types to varying extents. Tends to be the case that for more highly mutated cancers, signature 5 is also more prevalent in terms of number of mutations per megabase of sequenced genome, but it's there in all cancer types. What's also known about signature 5, and this was something interesting that we learned in reading the literature, is that it comprises about 75% of de novo mutations in children. So here's a study where parent-child trios were sequenced. So you sequenced a child and then you sequenced the two parents, and you ask, okay, what are the de novo mutations in the children? So these are the de novo mutations, and it turns out you can use the NMF to deconvolve this into 25% of signature 1. So remember, signature 1 is deamination of 5-methylcytosine, and 75% is signature 5. Okay, so this is happening all the time. And what's more, signature 5 tends to increase with age, but it's highly variable. So here's data from cancer patients. So we're looking at age of diagnosis for each cancer patient and their mutation load for signature 1 and mutation load for signature 5. So what you can see for signature 1 is that it's pretty well behaved. There's a straightforward relationship with age. Okay? For signature 5, there's usually a trend with age, but it's much more variable. So in fact, you have some data sets where the 95% confidence intervals are actually bigger than the graph itself. So there's a lot of variability with respect to signature 5 activity. Okay, so now we're going to spend the next bit of the talk talking about model system results, and this is from yeast work that our lab has done. Okay, so to make sense of this, I first need to introduce the system. So we're working with a haploid background that has the CDC13-1 temperature-sensitive mutation. And further, we have relocated 3 reporter genes, AD2, URA3, and CAN1, to a de novo, near a de novo telomere, so in a subtelomeric region. Okay. So what happens with this genotype is when we temperature shift it, the protective cap of the telomere falls apart and the DNA gets resected back. This exposes a long single-strand overhang. Now, if we apply DNA damage, what's going to happen is there will be lesions formed in this single-stranded region. So now if we restore the cells to permissive conditions, the DNA is resynthesized, but in the process, there's error-prone translesion DNA synthesis that makes mutations. So the net result of all this is this phenomenon called localized hypermutation, because the single-strand DNA is about 2 to 3 orders of magnitude more mutable than the equivalent double-strand DNA. And so we end up with multiple strand-coordinated, essentially simultaneous mutations. And we can detect these by plating on media with cannabinine to select for CAN1 loss of function, but with reduced adenine to select for AD2 loss of function because the AD2 mutants turn different shades of red. So we ask a simple question: what happens if we just let these cells accumulate DNA damage? What's that pattern? And this is what we get. So again, this is just shifted cells forming single-strand DNA, no added mutagen. And the pattern that we get, if we compare it to, again, cosmic signature 5, we get cosine similarity 0.933. So analogous to all the different species we looked at previously, we get a cosmic 5-like pattern. So first, we wanted to verify that this is indeed translesion DNA synthesis dependent, and it is. So here we're looking at CAN1 reporter inactivation frequency. So in wild type, we get this much mutagenesis, but if you delete either of the key translesion polymerases, then we get about order of magnitude less. So that's reassuring. So we looked into the relationship between translesion polymerase identity to the human polymerases as a function of cosine similarity when comparing the SNP patterns. So on this graph, on the vertical axis is cosine similarity versus human SNPs. And on the horizontal axis is amino acid identity to human Rev1. So this is one of the key translesion polymerases. And so a couple of things that are remarkable. One is that these species, indeed, you end up with cosine similarities, many of them above 0.9. But what's also very interesting is that even in highly diverged polymerases, some of them still produce high cosine similarity values. So this is something that, you know, we weren't sure quite what to expect going into this, but it was an interesting result. And analogously for Rev3, again, the amino acid identity here, we're looking at about 0.4, 0.5, and you still get high cosine similarity values when comparing against a human SNP pattern. So this gives you a sense of just how kind of deeply conserved this mechanism is on a molecular level. Okay, yeah, further experiments in yeast. So we were racking our brains thinking about, okay, how do we figure out what processes are required for this? So we tried a bunch of genotypes. So for example, you know, superoxide dismutase, catalase, etc., and none of them turned up anything. Then we thought, okay, maybe let's try manipulating the metabolism and see what happens. So this was a particularly telling experiment because in yeast, you can feed the yeast many different carbon sources and they'll grow just fine, they'll do their thing and live just fine. So we're looking at frequency of reporter gene inactivation. This is our mutagenesis assay. And we're looking at 23 degrees versus different lengths of temperature shift. So in orange here, this is dextrose or glucose. And here's a high level of mutagenesis. It turns out if you give the yeast a non-fermentable carbon source, so a carbon source that can only be utilized aerobically via the mitochondria, you actually get less of this mutagenesis. Okay, so this is acetate. And if you give the yeast carbon sources that has a mixed fermentative respirative metabolism, so these are galactose and raffinose, then you get mutagenesis that's actually somewhere in between. So glucose the highest, these kind of mixed metabolism sugars in the middle, and then the purely respiratory carbon source is the lowest mutagenesis. So then we thought, okay, well, is it glycolysis? How do we test this? Well, you can't simply knock out the key glycolysis enzymes because you'll either kill the cells or you'll make them very unhappy, but you can make a conditional knockdown using an auxin-induced degron. So auxin is a plant enzyme, and if you do your genetic constructions right, when you add the auxin, you can do conditional knockdown of a gene of interest. And the gene of interest for us is CDC19, which encodes the pyruvate decarboxylase at the very end of glycolysis. So if you knock it down, you're going to slow down the flux of glycolysis by a lot. And what happens when we did that is we dropped this mutagenesis by about an order of magnitude. So this was very interesting result for us. And what's more, we also checked whether the mitochondria are involved. So we took wild type cells and we made some of them into petites by using ethidium bromide to destroy the mitochondrial DNA. And it turns out for petites, it doesn't really make a big difference whether the mitochondria are there. So, sorry, day 0 is just cells at 23 degrees, so at permissive conditions. And then day 1, after shifting to 37 degrees, you see that there's not that big of a difference, so that mitochondria aren't a major player. Okay, and so I think this is the final experimental results slide. So this experiment, we thought, okay, let's just vary the glucose concentration. What happens if we use less glucose? What happens if we let the cells metabolize more glucose? So typically for yeast, the default concentration is 2% glucose, weight by volume. Okay, so at 23 degrees Celsius, we tried 0.5%, 1%, 2%, 4%, and 8%. And really, there's not that much happening at 23 degrees. That's not surprising because the cells are retaining their DNA in double-stranded form. When we temperature shift, that's when interesting things start to happen. So when cells are metabolizing glucose at 37 degrees, it turns out if you give the cells enough glucose to burn, you can actually induce more mutagenesis. Okay, and so, for example, if we compare 2% versus 8%, this is about roughly a 5-fold difference. So that's interesting. So then the next question was, okay, what about the actual mutations that are induced? When we actually sequence the cells from 2% versus 8%, and I should say these are matched cultures, so we would take a colony, suspend it in some water, and then split the water into 2 equal aliquots, one inoculate into 2%, one inoculate into 8%. So we did that, temperature shift the cells, collected mutants, sequenced them, and the 2 patterns are essentially the same. So cosine similarity of 0.971. So what this is saying is that 8% glucose, the cells acquire more mutations, but they acquire more of the same pattern of mutations. Okay, so just to wrap this up. So here's our working model so far. So we think that there's DNA damage happening just intrinsically from cells doing energy metabolism. And the indications from yeast so far is that glucose, for whatever reason, is especially mutagenic. And this DNA damage is acted on by translesion polymerases. And depending on the amino acid identity, so if we compare versus humans, depending on the amino acid identity, you'll end up with patterns that look like cosmic signature 5, but look more or less like cosmic signature 5, depending on how similar the translesion polymerases are. So this is the model that we're working with, and yeah. So obviously there are questions here that still remain to be answered. Okay, so just to quickly summarize, and I think I'm doing well on time. Okay, so many species have intrinsic base substitution patterns that all resemble this signature from cancers. So single base substitution signature 5, and they form what I like to think of as a continuum of related patterns. So they're not going to be just identical from species to species, but they're all going to be closely related, and this is revealed by doing comparisons like cosine similarity. So we think that the human form of this meta pattern, this pattern on top of patterns, is this cancer signature 5. And we think our results suggest that the mutagenic processes that give rise to signature 5 in cancers is probably the same thing as the intrinsic mutagenesis that's happening in just Darwinian evolution. That's giving us all these SNPs and all these variants in the model species that we saw. And the high similarity suggests, okay, there's probably conserved underlying biochemical mechanisms, which we're working to investigate. And the DNA damage from sugar metabolism is converted to a signature 5-like pattern in budding bees. So that's what we know so far. And just to wrap things up, future directions. So we're working to extend this experimental work to mammalian cells. The tricky part there is that culturing mammalian cells under standard conditions results in actually a lot of oxidative damage that's revealed by sequencing. There's a mutational signature for oxidative DNA damage, and that's signature 18. So if you're interested, you could look that up. So, the big question for us is, what is the actual DNA damage that's causing these signature 5-like patterns? Another big question is, to what extent is this actionable in cancers? If it were true that cancer signature 5 is related to sugar metabolism, then maybe all the folks doing clinical trials of caloric restriction and ketogenic diet, maybe they're onto something. It's also potentially possible that SBS5 is actionable by giving cancer patients inhibitors against translesion polymerases. Right, so what factors are responsible for differences in signature 5 occurrence? So again, as a function of age, it tends to increase a bit, but there's a lot of variability. So what are the factors that underlie that? And then finally here, can signature 5 prevalence be a partial predictor for cancer risk? So for example, people like to talk about liquid biopsies. Is liquid biopsy as part of an annual checkup going to become a thing in the future, where clinicians can sequence cell-free DNA and ask, okay, which mutational signatures are coming up? Should we keep an eye out for things? Yeah, that may be possible. Okay, and last but not least, I want to thank all the folks in my group, past and present. So Susanna's a postdoc, and Cassidy and Salma were former undergraduates in the lab, and together we have a manuscript that's currently under consideration at a journal for publication. And finally, I want to thank our funding sources for the ability to carry on this interesting research. And finally, I want to thank all of you for attending this webinar and for your attention. Merci beaucoup. And I'm happy to take any questions.
Thank you very much, Dr. Chan, for this seminar. That's very interesting. Some interesting patterns that you find in these cancer genomes. That's fascinating. Est-ce qu'on a des questions des gens de l'IBIS? Carla?
Yes, Carla.
Hi, it was very amazing, the talk. Thank you very much. I'm very interested in the last pattern. I didn't know about the glucose inducing mutagenesis. So I would like to know if we can have an idea of the level of bias we can have. For example, we are using another DNA damage, and we are also using glucose as media that could affect our results in mutation in our strains.
So are you talking about yeast?
Yeah.
Okay. You know what? I mean, this it seems this pattern is just so prevalent and it's so baked in to what cells are doing that it's basically quote unquote spontaneous background mutagenesis is the way I would interpret it. So it's also important to point out that these frequencies of gene inactivation, this is in single-stranded DNA. So typically, when folks are doing their yeast experiments, you can expect that your DNA is going to be in a double-stranded form. So yeah, so genomes should be pretty well protected. Yeah, I mean I guess maybe the only caveat to that I can think of right now is if you're doing experiments that does induce single-stranded DNA. So for example, folks who study replication stress, yeah, they're gonna end up with lots of single-stranded DNA. So they would need to kind of factor that into the way they interpret their data, probably.
Yeah, it was very interesting. I think I'm safe. I'm using diplates. But yeah, I have another question about this pattern could be like extend to human. So I was thinking maybe we are eating something that is causing DNA damage and could be extend to all the cancer that are the actual the actual harm of humans. So maybe glucose could be also causing that to human.
Yeah, well, so I mentioned the caloric restriction and ketogenic diets. It turns out if you go on clinicaltrials.gov, there are something like 35 or so clinical trials that are either ongoing or recruiting. And so, how are there so many clinical trials? It's because there's anecdotal evidence that at least for some cancer patients, if you put them on these low sugar diets, they tend to have better outcomes. So from what I've learned from talking to folks in the field, the conventional wisdom is that, well, sugar is, affecting, metabolism, it's affecting, TOR pathway, insulin, that sort of thing. And they attribute, potential benefits to that. But what we're finding is, maybe it's even more fundamental than that. And this gets back to a shift in my thinking that I used to think, okay, cancer is purely a genetic disease. If you pick up the mutations, in the wrong genes, you're going to have problems. You're going to get cancer. But what we're finding out with this metabolism thing is that, okay, maybe we should think about where those mutations are coming from in the first place. And so, yeah, it may be that, high sugar diets, especially, are bad for people dealing with cancers.
Thank you very much for the talk. It was amazing.
Was there another hand?
Yeah, Louis.
Yes.
T'es sur mute.
Yes, Louis.
On t'entend pas.
I'll say that again. Classic. So thank you very much for a great presentation, Dr. Chan. And I have many questions, but thankfully we're going to meet at 2 o'clock. So I'll say some for the discussion. I guess if we can take another question, Christian, I just need to go to the door to pick up the exam copy. I'll be right back in a minute. Sorry.
Yeah. Est-ce que vous avez d'autres questions? I have, yep.
Oh, good. You can hear me. Thank you very much for the talk. It was very interesting. I was wondering, with the data that you're showing, you kind of, it seems like you average across the entire genome for the substitutions you observe. But do you see like specific signatures within coding regions of the genome or non-coding regions of the genome? And do you see any effect of the, I'm not sure I understood if the measurements that you presented were kind of normalized for the differences in GC content between the different species for which you showed data. So I'm just curious how this can vary along the length of the genome.
Right. Yeah. So that's a very good question. So these are, well, let's see. So we took the most basic approach. So basically not trying to apply these sorts of corrections. And so I guess the most basic reason for that is that if you see the similarity without applying those sorts of corrections for the specific frequencies of specific nucleotides, then I think the argument is in some sense stronger. Having said that, though, the question that you asked, especially related to whether there's a transcriptional bias. So for signature 5, from the cancer data, there is no significant bias. Having said that, we have not systematically gone back through all, 50 species and looked into that. That's something that would be great to do if I can convince somebody to kind of take over this project full time, because I'm still doing a lot of the computational work myself. So we wouldn't expect that there would be a strong transcriptional bias, but the answer is we haven't actually had the chance to look yet.
Thanks a lot.
Louis, veux-tu y aller avec ta question?
Yeah, it's probably still the time to ask it anyway. Well, you're probably gonna destroy my question right away, but I was kind of wondering why I mean, your main target is SBS5 signature, actually. Like in the comparison with all, basically what you're showing is that this pattern is the most prevalent, probably the most tolerable and the most expected under neutral, not neutral expectation. And to me, it seems that other signatures that depart from this and are more specific to different cancer types are the most interesting and perhaps the most consequential in terms of functional impact of the related to, let's say, to the severity of the cancer or the probability of relapse to evolve resistance to treatment and so on. So I'd like you to comment on that.
Right. Yeah. So there are, I think, two. Yeah. So I think there are kind of two schools of thought on this. So there is a thinking that, okay, if this is just kind of background spontaneous mutagenesis, it's going to happen anyway. So that's the thinking. So wouldn't it be more interesting to focus on things that are, more specific to a particular cancer type? So, for example, signature 7 with respect to, skin cancers. So that's clearly actionable. Let's see, what else? Well, signature 1 is not actionable. You can't do anything about that. It's just biochemistry. Signature 2 and 13, these are ABABEC activities. So there are groups and I think even companies working on ABABEC inhibitors. Homologous recombination, there are drugs out there, so PARP inhibitors that try to limit the genomic damage from homologous recombination deficiency. So you're right. Yeah, there's a lot of interest in just signatures all around, and there are people definitely pursuing many tracks of research and development along the lines that you suggest. but I think the case that I would make for signature 5 though is that there are people, there are people interested in it. There's speculation in the field as to where it's coming from, and does it matter? It seems to be everywhere, and I would argue that if it is everywhere, and especially in some cancers where it may actually comprise the majority fraction, or at least half of the somatic mutations, if it's something that's actionable, we should try to do something about it, because it's important to realize that signature 5, and in fact many of these signatures, they remain active post-diagnosis. So they remain active while the cancers undergo subclonal diversification, so producing more and more genetic heterogeneity. And this genetic heterogeneity causes problems for treatment, because the more diverse the tumor, the more likely something resistant is going to emerge and become a subclone and start taking over. So not to worry, there are plenty of people working on other signatures for sure. Yeah.
Okay, thank you.
Gabrielle?
Yes, thank you. Really enjoy your talk. It was great. Thank you. So I had just 2 questions. The first one is, that signature you propose it's linked to metabolism. That would be the driver behind it. So I'm not a yeast guy, but from what I remember, metabolism is glycolysis is driven by oxygen concentrations, right? So fermentation versus aerobic utilization of glucose. So my question is oxygen part of your model or your thinking?
It is, in the sense that you're right that the most glycolytic tumors tend to be very hypoxic, and so there's kind of this inverse relationship going on. As far as the yeast like to ferment whether oxygen is around or not. So in that sense, it's advantageous to study glycolytic metabolism in yeast. So in terms of the inverse relationship of oxygenation versus glycolytic activity, this is something that we're taking into account with the mammalian cell culture work that we're pioneering. And what we're trying to do is we're trying to culture our cells 100% of the time, all the way up until DNA extraction in a glove box, so maintained in low oxygen. Because what other labs have done is they've used low oxygen incubators, but when they're splitting their cells, they're just taking them out of those low oxygen incubators, taking them to room air oxygen, and doing, feeding their cells, splitting them, and then shifting them back. So what happens is that you're going to get spikes of oxygen, high oxygen. Yeah. And so, you're absolutely right. We need to definitely take into account. And what I would also point out about the oxygenation and glycolysis in cancers is that there are some nice papers looking at in vivo imaging of glucose metabolism, and also of factors like intracellular pH. And when the systems are set up right, you can actually see gradients within tumors. Now these are model tumors, so these are HeLa xenografts in nude mice. So one can argue, okay, does that really reflect real tumors? But there's literature out there looking at that, looking at oxygenation and glycolysis.
Yeah, thanks. One last question. So that manuscript, can you tell what's the journal it's been submitted to, on the review?
Does that matter?
No, just no, but I would assume it would be pretty good impact, right? Because your stuff is pretty novel, right?
Well, you know, it's it's a fairly high profile journal, but the thing is with peer review
never know.
process,
Yeah,
you never know. If you draw a reviewer who's just upset for whatever reason, you know, better luck next time at a different journal.
Well, that's what I wanted to tell you. So good luck with your manuscript, and looking forward to read it once it's published. So best of luck.
Thank you very much.
All right, thank you. Maybe we should stop here since it's 1:30. And merci tout le monde d'être venu. And thanks again, Dr. Chan, for your seminar. And please stay online for the individual meetings.
Okay.
Thanks, everyone.
Thank you all very much. Thank you.